switching gears to RNA-seq data analysis, and Matthew Gazzara is going to tell us about a pipeline to detect, quantify, analyze both classical and complex splicing variations from RNA-seq data.
Hi, thanks for coming out to listen to my talk. I'm part of the Barash Lab at Penn, and Yosef just introduced my talk, so let's get right into it. So our goal here is to define and quantify RNA splicing variations given RNA-seq data. And given Sebastian's nice introduction to all this, I'm going to skip over the formalities of why it's important to look at splicing. But in our tool, we want to have robust and reproducible quantification that's scalable for large data sets, relatively fast for large data sets, but also importantly, easy to interpret and user-friendly for users who don't like parsing tab delimited or binary files. So our solution is the LSV formulation and our algorithm MAJIQ. So first, let me tell you what an LSV is. An LSV stands for a local splicing variation. So it's a graph-based definition for alternative splicing events. The nodes here are exons, reference exons, and the edges are junctions that connect those exons. So this leads to two basic LSV types. You have single source LSVs where you have a reference exon with junctions emanating out of it to downstream 3' splice sites, or target LSVs where you have junctions or edges coming into an exon from upstream 5' splice sites. So this formulation allows us to get your standard set of classical splicing events that are binary decisions, but importantly, it also allows us to capture more complex splicing variations that involve 3 or more junctions. So we've analyzed a lot of data sets, and it turns out that complex LSVs are very prominent, more prominent than you might expect. 30% of all splicing variations in the transcriptome have 3 or more junctions. That constitutes 50% of regulated alternative exons, and these exons are more highly conserved, even compared to binary alternative splicing events. So we think they're important for you to capture properly. If you're not using MAJIQ or a tool that handles complexity like this, you're either missing events or misclassifying them as several binary events. So this is an example of the gene EIFG3, 4G3. This is a simplified splice graph, and you can see lots of exons, lots of junctions going all over the place, but importantly there's a lot of green things here. Novel exons detected, novel intron retentions detected, and novel connections between the exons. So that's a unique thing that MAJIQ does also, all this novel detection. So we could zoom in on this single source LSV, lots of junctions, lots of variable exons, and MAJIQ accurately defines what isoforms we would expect to see when we do RT-PCR in these mouse tissues, and it gets the quantification of these complex isoform landscape of this single event very accurately. So this is delta psi by RT-PCR, and what MAJIQ predicts for for a handful of events across 11 different tissues. We've got nice accuracy there. So we wanted to benchmark against other tools commonly used by the field, so we have to restrict analysis to binary events because they don't handle complex LSVs. So basically we did over 200 RT-PCRs and compare psi and delta psi measurements, and we compare very favorably to other methods. As another kind of high throughput way to assess how reproducible our data is, we've developed this reproducibility ratio here. So just to give you an idea of what this is showing, the idea is you have two experimental conditions and you want to find out what exons are changing or what events are changing between those two conditions. So you run an experiment where you pick replicates from your two conditions, find your high confidence set of splicing changes, and then you repeat the experiment, except now you're using a different subset of replicates. And you say, okay, given my second experiment, how many events were reproduced by in the first experiment? We're just using combinations of replicates to see how reproducible we are. And MAJIQ in blue here, compared to other methods, has higher reproducibility ratios Perfect reproducibility is along the 45, and the reproducibility ratio is the endpoint here. We achieve 86% reproducibility. Also importantly, you'll note for sensitivity, we are able to detect more splicing changes than other methods. Now it's important to, because I mentioned earlier we can handle Denovo junction detection while other tools can't, we repeated the analysis with removing Denovo junction detection. And you can see that from non-Denovo to Denovo, you get 30% more splicing events without any reduction in reproducibility. Kind of piggybacking on what Sebastian said, there's a lot of Denovo unannotated exons out there. And this is comparing two normal mouth tissues. We're not knocking down splicing factors, looking across all brain development, or looking at cancer or some sort of crazy disease. So if you're not using Denovo junction detection, then you're going to be missing probably more than this. It's a lower bounds. So again, we want MAJIQ to be a user-friendly option if you don't want to look through a tab, delimited file, or parse a binary file. We have interactive HTML5 outputs that you can view in Chrome or a browser that make it a bit more accessible when you want to explore what's happening in my data set. So this is the index file, kind of portal for your analysis. You can sort and apply filters to look at specific LSVs. So here we've sorted for the most changing. It's comparing embryonic to adult. Cortex is actually the data set Sebastian just talked about. And we have our most changing LSVs here. We could view all of them, do Control F, search for your favorite gene of interest, etc. If we click on the link here, we get a more detailed view of what's happening at a specific, for a specific splicing event. So here I'm showing splice graphs for adult and embryonic brains for the splicing factor PTB protein 1, which is downregulated over development. And we see that MAJIQ detects a novel poison exon over brain development that is induced in adult brains. So you can display the posterior distributions for psi, delta psi for your different conditions, get an idea of what the spread looks like there. But also we have links to UCSC where you can have your favorite tracks, custom tracks uploaded, and you can get an idea of exactly what's happening within the bounds of this LSV that we detect is changing. Very clearly the exon is induced upon cortex development. So MAJIQ, I told you all these great things, but how does it work? There's 3 easy steps that on the command line don't look too intimidating. So if you're not computational savvy, I think you could still get this to work. You define the LSVs, you quantify the LSVs, and you visualize the LSVs. We do this in 3 steps also because you can parse out the different steps of the pipeline. The first step is the most computationally heavy, so you probably want to run that on your desktop computer or your HPC, but I'll show you, we've made recent developments where even this step can be run on your laptop computer. So that's the basic pipeline. So we really want MAJIQ to be usable for a lot of different experimental needs and contexts. So all you have to feed MAJIQ is sorted index BAM files mapped by your favorite aligner, and an annotation database of genes, and we'll look, define the LSVs, detect Denovo junctions, etc. To speed things up, if you only care about a subset of genes for your disease or condition that you're looking at, just feed us a smaller database and it'll go much quicker. We can handle strand-specific libraries. If you want to analyze old data with new data that has different read lengths, short or single versus paired end reads, we can handle mixed library prep conditions. If you want to save time, you can run non-Denovo options. If you want to simplify things, you can look for LSVs and junctions that have support in your data only. And lastly, real quick, we also handle replicates, as I mentioned. Lastly, real quick, I want to talk about 2 things we've been developing more recently for scalability and outlier detection in large heterogeneous data sets. So Jordy, the developer in the lab who wrote MAJIQ, re-implemented MAJIQ using HDF5, and you can see now that it's very scalable with the amount of memory it uses per CPU. under 500 for this range that we see here. So now we can run really large data sets for splicing quantification. It's 6 times faster than the old one. This implementation can be implemented on HPCs and other highly distributed environments. And importantly, we can run a fraction of the GTEx consortium of over 1,500 RNA-seq experiments in a reasonable amount of time. On Yosef's desktop, using 32 threads, it took about 4 days. On Penn's HPC, it took 7 hours, so very reasonable. And even with this large amount of samples, we're at 600 megabytes per process. And finally, I just want to touch on work from our graduate student Scott in our lab. He's working on outlier detection. So obviously, this is useful when you have heterogeneous data sets where you have differences between patients that is real and or confounding. So you can see here, we take a set of LSVs where we perturb the splicing pattern in one of them, make condition an outlier, and we can ramp up how many, what fraction of those events are perturbed, and you can see without correcting for this, the number of changing things skyrockets and the reproducibility plummets very quickly. But it's hard to see here, but with his weighted model, you basically recover as if the outlier was removed, and you have all good, nice looking samples. So we think this is applicable for patient data. So last, acknowledgements. This was a group effort with Jordy and Alex in the lab. Jordy wrote the algorithm, Alex did all the visualization. I did downstream analysis and mixed clear liquids in tubes for experimental validations. So check out our website. We have a Google group. If you have comments, suggestions, questions, we're really anxious to answer and implement what you need to do your analysis. And more details are in the paper cited here, so thanks.
Yes.
For the Denovo splicing detection, what kind of algorithm?
What kind of algorithm?
Yeah, what kind of algorithm?
So,
Matt, can you repeat the question?
he was asking for Denovo detection, what kind of algorithm do we use to detect those junctions? So MAJIQ doesn't find the junctions, your aligner will have to do that. MAJIQ finds those novel connections in the index BAM file you give it. So we use STAR, so if STAR finds a novel connection and reports it, we don't just look at what the database said should be there, we look for new connections and analyze them. So the aligner is really what's detecting them. MAJIQ just is aware of that and analyzes it downstream.
Yes.
So I'm just wondering, because you mentioned the psi values, so when you're running comparison analysis between 2 different cases,
Right.
what would be your recommendation for comparing the psi values to find the significantly upregulated or downregulated, alternatively splicing events, basically?
Right, so the quantifier part is really 2 processes. We have psi, if you want to look at within a condition. We have a completely different process called delta psi. It's a different command that runs and outputs exactly what you want. It calculates psi, puts them together with normalizations and corrections in a prior, and it outputs things that are changing given your threshold of interest, 10%, 20%, show everything. So it's a separate execution that reports the psi for your 2 conditions and a delta psi value.
So it wouldn't be biased in favor of the genes that have less number of alternative splicing events, is it? So let's say that a gene has like a 10 alternative splicing events with operator gene.
Mm-hmm.
So this psi values is basically the fraction of the reads that are basically assigned to each event.
So it's each
transcript,
junction.
technically.
Each junction and the events are exactly the same in the 2 conditions, right? So you're comparing junction by junction by junction. And what's nice about delta, it's a direct comparison between 2 conditions. So things like that go away when you do the delta.
One last question, and then we can do a discussion. Sorry. Yeah.
So, since you've thought about a lot about these very complex splicing events,
Yes.
what would you suggest to the downstream analysis and the interpretability of the splicing events. So you said exon is very easy to interpret.
Sure.
You now have this new piece of sequence. How does that change the protein? How would you suggest going about interpreting the downstream effect of these complex splicing events?
Right, so it is a challenge. In the paper, we mapped protein domains to these different LSVs, but it's hard to say, like, we don't currently have something implemented that says, like, how does it change the reading frame? It's all annotation based. The truth is, when you look between two conditions, you tend to have two junctions changing, and maybe the third one changes a little bit. It's when you start looking at multiple comparisons together that you really see usage of 3 or more junctions a lot. So if you want things to be simple, we have the data only option where you'll get simple things. But the visualization is really key. So it's hard to make sense of complex LSVs all over the place, but looking at the splice graph output, it makes sense.
Is that possible with the MAJIQ psi option without delta psi?
Yeah, so the output's basically the same. You get splice graphs and psi
We can
posteriors.
talk more in the discussion, otherwise we're going to